It's me, Rima Boripan, and we have to go to the lecture 4 of the clinical chemistry. So, we are talking here about what is the difference between the physiological and the pathological. So, here is the physiological, that the RBC is discarded by the spleen. Okay, because we know that is physiological when the RBCs are old enough, and we know that the lifespan of the RBCs it will be or it is 120 days. It's about 3 months, so they are decarded by the spleen. But what is so pathological that the RBCs decarded in the liver and the bone marrow, that is not normal anymore. So if the RBC is discarded by the spleen, this is normal. But if the RBC is discarded by the liver or the bone marrow, that is not normal. Now we just make a table to differentiate between what is increased in the intravascular hemolysis and what is increased in the artificial hemolysis. So we have here intravascular hemolysis, lactate dehydrogenase, indirect bilirubin, reticulocytes, free hemoglobin in the urine. So first we will talk about the lactate dehydrogenase. So we know that the lactate dehydrogenase that is present in the systole of the RBCs. So intravascular hemolysis, that's mean it happens inside the vessels, inside the organism. So actually it's present inside the cell. So it will be an increase in the both sides, if it's intravascular hemolysis or if it's artificial hemolysis. Indirect bilirubin, the RBCs should be discarded and they changed into the inside the spleen, into the heme, more protoferrin, and then to the unconjugated bilirubin. So it needs spleen. So that is not an increase in the artificial, it decreased actually. And the reticulocytes, they are immature RBCs, which they are created from the bone marrow, so also decreased in the artificial hemolysis. Free hemoglobin in the urine, not reliable markers to distinguish between them, so we can distinguish them by the free hemoglobin that is present in the urine. So if I wanted just to compare between two things, between what are the things that is an increases in intravascular hemolysis and what is not an increase in the artificial hemolysis, what is an increase in the intravascular hemolysis, the first thing is bilirubin, lactate dehydrogenase increase in both them is the only thing that increase in the both of them, but the unconjugated bilirubin and also the reticulocytes increased only in the intravascular hemolysis. Now we go to the lactate dehydrogenase enzyme. Lactate dehydrogenase produced after breakdown of the RBCs. Once we know that the lactate dehydrogenase enzyme that is present in the systole inside these cells, and the common type that is associated with the RBCs is called lactate dehydrogenase type 2. Okay, that is produced after the breakdown of the RBCs. Indirect bilirubin is the breakdown of the heme by the spleen. In the artificial hemolysis, no spleen, so it decreased, not constant, it decreased also. So that is explain why the indirect bilirubin it's not an increase or it's decreased in the case of artificial hemolysis, because in the breakdown of the RBCs that happens inside the spleen, it will turn to the unconjugated hemoglobin, but in the artificial hemolysis there is no spleen, so that is lead to decreased amount of the indirect bilirubin. Now we go to the reticulocytosis. Reticulocytes are the early stage of the RBCs. That's why I said immature RBCs. There is no bone marrow to reproduce them, so also decreased in the case of the artificial hemolysis. Now we go to the protein is called haptoglobin. is the most sensitive marker of the intravascular hemolysis because we are talking about the intravascular hemolysis. Let's talk about the information that is present here about the haptoglobin, okay, and then maybe I will talk about other informations about the haptoglobin. So first information or the first note that I wrote here is the haptoglobin is the most sensitive marker of the intravascular hemolysis. Despite in the intravascular hemolysis an increase of the lactate dehydrogenase, unconjugated bilirubin, reticulocytosis also an increase, potassium level an increased in the intravascular hemolysis. So many things that is an increase in the intravascular hemolysis. Despite all these things, the most sensitive part is haptoglobin. But the question is, what is the haptoglobin? Haptoglobin, it's globin, you can see, so it's a part of a globin. And the globin, which type of a globin is alpha 2 globin? It's actually the haptoglobin, it's alpha 2 globin. So just memorize it. It's not any globin, it's just alpha 2 globin. Is a plasma protein that is binds to the free hemoglobin, inhibit the release of iron from them. Haptoglobin doesn't bind to the RBCs, it binds to the free hemoglobin. Okay, why it's bind to the free hemoglobin? To prevent the release of the iron. Why? Because iron causes oxidative damage. Okay, so by that preventing the oxidative damage that causes damage to our body. Where it's synthesized in the liver, it's actually plasma protein. Most of the proteins they are synthesized in the liver. Haptoglobin only binds to the 1% of the hemoglobin in the erythrocyte at the usual hematocrit. That is normally okay, because there will be a free hemoglobin, but it just bind to the 1% of them. That is what is normally, and it will be an increasing in the acute phase protein. So again, haptoglobin is alpha 2 globin. It's a plasma protein synthesized in the liver and bind to the free hemoglobin that is preventing the release of the iron from this hemoglobin. Why? Because that is prevent the oxidative damage that is maybe caused by the iron. Normally, it binds to the 1% of hemoglobin in the normal hematocrit, and we just discussed before what is the hematocrit. Hematocrit is a percentage, and we can measure it by measure the height of the RBCs divided by the height of whole blood sample multiply 100, or we can measure it MCV multiply of the RBCs multiply of 10. And we said that normally it's 45%. We discussed all these things about the hematocrit, and that is an increases in the acute phase protein. As we can see here, this is the haptoglobin bind to the free hemoglobin inside the macrophages, because the macrophages take this hem, okay, and then go to the spleen, and then that is lead to the production of the indirect bilirubin. What's so important, yes, is a sensitive marker, and that's not mean exactly that it will be an increase. No, it decreased in the intravascular hemolysis, because intravascular hemolysis lead to the breakdown of the RBCs, release of the hemoglobin, then it will be free hemoglobin. Once it can, actually there is normal haptoglobin in a normal value, but the haptoglobin bind to the hemoglobin, so we can see the hemoglobin, and then quickly these macrophages take this complex and send it to the spleen. That is the mechanism. I will talk about other information about this, and maybe something will be repeated. So again, haptoglobin, it's a plasma protein, it's alpha 2 globulin. What is happen? Hemolysis, free hemoglobin, haptoglobin make with the free hemoglobin complex, and then the macrophages quickly take this complex and eat it and send it into the spleen, so that decrease the level of the haptoglobin. Yes, it's sensitive mark, but that is lead to decrease the level of the haptoglobin. The other thing that is synthesized in the liver, we talk about this. Hemolytic anemia destroys of the RBCs, and then the hemoglobin release make it a complex. Macrophages eat it quickly and then send to the spleen, so that is lead to decrease the haptoglobin. What so special about the hemolytic anemia? I'm not talking about the intravascular hemolysis because it's another topic. I'm talking about the hemolytic anemia, and we know that the hemolytic anemia, both types happened: intravascular hemolysis and extravascular hemolysis. About the intravascular hemolysis, because the macrophages, they are present, they are just directly take this complex and eat it, so that is lead to decrease the presence of the haptoglobin in the blood. But when I'm talking about the extravascular, that is lead to the destruction in the spleen and some escape to the plasma. That's why we can watch decrease in the haptoglobin in the intravascular more than the extravascular, because the extravascular, the destruction will happen in the spleen more than in the plasma, but some will escape to the plasma, so the percentage will be not right. Okay, it's a specific protein bind to the free hemoglobin, not any protein that is lead to these conditions. Okay, free hemoglobin increase, so that is lead to decrease haptoglobin because they are bound together. Haptoglobin synthesized in the liver, and the normal level of it will be 30 to the 200 milligram per deciliter. So this is the normal level. What we can see in the intravascular hemolysis decrease, that's mean it will be less than the 30 milligram per deciliter. What else? Also, so we can see free iron, and that is can stimulate infection. Consider haptoglobin in the acute phase reacting. So this complex, I can see that this spleen and macrophages, both them that eat it, so that is lead to decrease the haptoglobin. About the iron, when I say that oxidase damage to the blood, to the body, that's mean exactly that is causes enhance the infection. Okay. mm, there some said about this, because we are said that is alpha 2 globulin, but actually some said that the haptoglobin is a tetrametric structure that is composed of 2 alpha 2 beta units, and they are connected together by the disulfide bond. Why it decreases during the hemolytic anemia, or we can say intravascular hemolysis, because it used or it used up faster than the liver can produce it. So in the liver disease, when it's in increased acute phase reactant, that is lead to decrease the level of the haptoglobin. So here we have two conditions, like some said that is called 2 alpha globulin, but it's actually globulin. Okay, and some said that is a tetrametric structure that is composed of 2 alpha and 2 beta, and these units, they are connected together by the disulfide points. Now we go to other topic, which is protein electrophoresis. And protein electrophoresis, actually it's the measurement of the charged particles in an electric field. We make it like a charge and we measure these particles, and that depends on the charge, the size, and the shape. What is the charge of this particle, the shape of this particle, the size of this particle? So they will be separated, and from its name actually we can say it's a protein electrophoresis. Electro, that's mean we use electrical field. Phoresis is a Latin word that's mean separations of these protein. So it separates the components of the plasma protein with a negative charge. So the plasma proteins, all of them, it will be the negative charge, Okay, and then separated them. So that's not depending on the charge of all these plasma proteins. I make them all the negative charge because we have several types of it. So the separation will not depend on the charge of it. Okay, so we can say here the negative charge, here the positive charge. Most of the plasma protein negative charge, we put the buffer and using a CAF, cellulose acetate film, and not the gel. Here in the protein electrophoresis, here we use cellulose acetate in the buffer. Cellulose acetate, this is the CAF. Okay, here we have the negative charge and here we have the positive charge. Most of the plasma proteins, so we just control the conditions like this buffer and also the temperature and other things. So actually it will be these plasma proteins will go in these directions because they will be negative. We didn't use gel, we use a cellulose acetate. We use serum and not the plasma because why? In the plasma there is a fibrinogen which affect protein concentration because it's a protein also, and it's acute reaction protein. That's it. So in these conditions we use actually the serum and not the plasma. Because why we use the serum and not the plasma in this condition? Because we want to measure the protein. Okay, and in the plasma there is a protein called fibrinogen, and that is make like interference with the other proteins, so I can't get or get a good and a correct results from that. So that is the protein electrophoresis. Again, we use the serum, we can't use the plasma because the plasma contains fibrinogen, which make like manipulations of the results and not the good and well results. So screening and monitoring of the plasma proteins. Actually, before I go to this part, I want to explain more and discuss all the types before I discuss all these things that is present here. So the first thing that I said before, that most of the plasma proteins contains the negative charge, and we will describe the things. There will be something that we will discuss it actually. um, Other things is about the plasma proteins. We wanted to know all the plasma proteins, like here we set these reference intervals. We want to know what is the albumin, alpha 1 globulin, alpha 2 globulin, beta 2 globulin, alpha 2 globulin. Actually, we said that the haptoglobin is alpha 2 globulin, but another source that said that is composed of 2 alpha 2 beta, and they are connected together by the disulfide bond. The most important resource that is said that's alpha 2 globulin. So about the first one is the albumin, and we can see here that the albumin is the highest percentage, it's about the 70 percentage, but we want to know more information about what is the albumin. Actually, total plasma proteins, as we see here, it composed of the albumin and globulin. And what do you think, which one of them is the more concentration? Albumin is more concentration than globulin. Albumin is 4 milligram per deciliter, while globulin is 3 milligram per deciliters, and the total it's 7 milligram per deciliter. So the total plasma protein is 7 milligram per deciliter, but we have albumin, we have a globulin, and the albumin is more than the globulin despite all these types of a globulin, but the albumin is more concentrated than the globulin, not high percentage, but it's more. Increase the level, we can say, when the albumin increases and when it decreases. Okay, so that's about the albumin. That's the important thing that I want to discuss it now. So we go to some said that serum protein electrophoresis. You see what I you hear what I said? Serum protein electrophoresis. So we didn't use the plasma. We don't use the plasma because it contains the fibrinogen. So it's a quantitative, not qualitative measurements. Quantitative, that's mean it measure the quantity of it and not the quality of it, because we know an increased number of immunoglobulin, but we actually don't know which immunoglobulin that's like it measures. Okay, there will be an increased level of immunoglobulin, but it can't be measure the which immunoglobulin that is increased. So we have albumin 70% and gamma globulin 19%, which is antibodies, beta globulin 13, alpha 2 10, and the least is alpha 1 globulin. This drawing is so important, really. It's so important to know what is the normal. As can we see, this is the albumin, and it will be normally it will be the highest, and as you see the gamma globulin, it will be the second one, and then the beta globulin, then alpha 2, then alpha 1 globulin. Okay, why is the albumin is the highest? Because the albumin large protein. Okay, and when it's more large protein, more negative charge. Okay, so more large protein, more negative charge, so it will be the nearest to the anode, and that's why it takes the peak, why the albumin is the largest here. Okay, albumin is more abundant, and as we see it, the albumin is large protein, so we can say alpha is subdivided into alpha 1, alpha 2. Alpha 2 globulin is haptoglobin, and beta globulin, we have examples of it like transferrin and coagulation factors. They are beta globulin, so the beta globulin, so we have albumin. Okay, we understand albumin is the larger protein that this contains negative charge, and the gamma globulin is the second one that responsible for immunoglobulins and antibodies. I mean, the beta globulin is the second, the like a transferrin, like a coagulation factors, then we call them as the beta globulin. Okay, and that's all that I can here describe it. So first I will see here in this table what makes albumin increases. The only thing that make the albumin increases, maybe that is not present here, but to make albumin increase more and more, that's the dehydration. So dehydration causes more albumin. Okay, so dehydrations increases the level of the albumin, but what reduces it, the other causes like malnutrition, malabsorption, nephrotic syndrome, as you see here, inflammatory conditions, you know, um, vascular morbidity, overhydration, pregnancy, liver cirrhosis. So all decrease the albumin. The only thing that increase the albumin concentrations is dehydration. Now we go to the alpha globulin, because we know that the alpha 1 globulin is the smallest, and when you see it's larger like this, so that's mean many reasons for that. Inflammatory condition, whatever is it, so nephrotic syndrome increases. This is the only thing that is, but it's increases also in the inflammatory, but it's in acute inflammation. Okay, it's not increases in the acute condition. So alpha 1 increases in the nephrotic syndrome. The only thing that is increases is nephrotic syndrome. Alpha 2, the only increases in nephrotic syndrome and the tumor. Look, that is an increases in a nephrotic and the tumor, acute inflammation, that increases the alpha 2. Beta globulin like haptoglobin, so the haptoglobin like alpha 2 globulin, that decrease in the intravascular hemolysis but increase in the nephrotic syndrome, tumor, acute inflammation, alpha 2 and nephrotic syndrome. And beta globulin, an increase also in the nephrotic syndrome, an increase in the paraproteinemia, an increase in the liver cirrhosis. And the gamma, an increase in chronic inflammation and the liver cirrhosis, an increase in the paraproteinemia, but decreased in the anticorpor deficiency. So as you can here see, the albumin is only increased in the dehydration, decreased in the nephrotic syndrome and liver cirrhosis, alpha 1 increase in the nephrotic syndrome, beta alpha 2 also increase in nephrotic syndrome, beta increase in the nephrotic syndrome, and gamma increases in many conditions. Beta, you can see that's an increased nephrotic, paracytonemia and liver cirrhosis. Okay, so we discussed before that about the influencing factors, and I talk about the vitro influencing factors, and one of them was prolonged hemolysis. In the case of a prolonged hemolysis, that's mean in a prolonged tourniquet use, and that is lead to the prolonged hemolysis, is also this condition that also increase the protein level. Now I will discuss the other things which is known as SDS page, and that's so important to discuss it. Actually, it contains a lot of information, but first we will discuss all the information that is present in the PDF that is in front of us, and then we will discuss if there is other information that as I can talk about it. So here we have, as you can see here, we have this box. I can see we can see here the first one is cathode, which is a negative charge, and we have also here the anode. Okay, so actually before we discuss, this is the stacking gel, this is separating gel, and this is the buffer, and here I said this is a plastic film, and I'm talking about the pH. Here we have here is glycine, here is gluconate, here the pH is 6.8, here the pH 8.8. so as you can see here that the SDS, what is the name of the SDS? S sodium, D dodecyl, S sulfate, and then we talk about the page, which is polyacrylamyl gel electrophoresis. So this is the whole name of the SDS page. So S sodium dodecyl sulfate polyacrylamide gel electrophoresis. So we now know that is electrophoresis and used for the separations of the protein. Okay, so we have here a negative, we have a positive, here the stacking gel, here is the running gel, and there is a buffer. And you see that in the stacking gel we put the glycine, and the glycine, the pH is 6.8, which more acidic. But here in the separating gel, we can say running gel or whatever, the pH is 8.8 and is contain glycinate, which is, more alkaline. Okay, about the buffer is lamelli buffer. The question is why we put the sodium dodecyl sulfate? Because it causes denaturation of the proteins by binding to the hydrophobic regions. We will discuss also why it makes the SDS denaturation of the protein, but like a general idea, what is the purpose of the SDS? The purpose of the SDS that is causes denaturation of the protein. Why we causes it denaturation of the protein? Because we want all the proteins will be a negative charge. We want like all of these proteins has the same charge, so which is negative, so these proteins can be just migrating from the negative side, which is cathode, and migrating towards the anode. That's exactly why we cause denaturations of this protein. So we think about the SDS. So the sodium diethyl sulfate causes denaturation of proteins by binding to its hydrophobic regions. Okay, so like bind to the non-covalent bonds and then break it. So what is so special, as I write here, anionic deter agents denaturate the proteins to their primary structures. They give it negative charge. That's it. So the sodium diethyl sulfate anionic deter agent causes denaturations of a protein and give it all of them negative charge by binding to the hydrophobic regions of these proteins and causes all of them contains the negative charge. Then there is something is called beta mercaptoethanol. We added it to this. Why? Because sodium diethyl sulfate, okay, causes denaturation of proteins, causes all of the proteins contains the negative charge, but doesn't solve the problem of the disulfide bonds. Maybe some protein they contain the disulfide bond. Why denaturation of a protein? Because some proteins are like folded on something, like make it defolding of these proteins, also make them as the same size, same charge. But some proteins contains disulfide bonds, so we add beta mercaptoethanol to break these disulfide bonds. So what is the purpose of the beta mercaptoethanol? Reduces the disulfide bonds, especially when it's spoiled. It's a reducing agent, it's not anionic reducing agents, and causes a breakdown of the disulfide bonds. So the proteins now, they contain all the negative charge and all the same size, and they just runs or migrate towards the anode. So what is the difference now is the molecular weight. So they are not separating according to the size or according to the charge or according to anything. They are just separating according to the molecular size, because we add SDS and we add beta mercaptoethanol to make all the proteins have the same size, the same charge, so they can migrating depending on the molecular weight. So the migrating or the arrangement of these proteins in the SDS page, that is depend on the molecular weight of them and not their charge or something. Okay, that's important. Now we talk about the glycerol. Okay, the glycerol increases the sample density and viscosity. It increases the density so we can see it. Okay, and there is something is called bromophenol blue. Why we use the bromophenol blue? Because negatively charged dye blue at the pH more than 4.6, and that's why it's 6 and 8, because it should be blue because we want to see these proteins. Okay, without this dye we can see any proteins and we can see the migration. So bromophenol blue, it's a negatively charged dye, which is terran negative, or which is terran blue, I mean, when it's in the pH is more than 4.6. Allows for monitoring for the migration. It gives the sample blue color, so we can detect it. You're just monitoring it by seeing what is the blue color, so we can say bromophenol blue. So we have a bromophenol blue, we have the glycerol that is increasing the density, and the distilled water, because we don't need ions in the sample. That's why we use the distilled water. So now I will repeat some of them again. Okay, so I don't want just to repeat everything. I just focus on the glycerol because I didn't talk any too much about it. Glycerol increase the density of this sample. Bromophenol blue, that's an important from its name, that is give a blue color when it's the pH is below the 4.6, and the distilled water because we don't need any ions inside it. As you can see, we have a discontinuous gel system because we said here there is a stacking gel and separating gel, so this is a discontinuous gel. We in the stacking gel the pH is 6.8, so we give HCl and tris. It gives Cl for the SDS for their reaction, so we give the Cl, and then the running the pH is 8.8. Okay, running puffer, tris, glycine, as I said, and the SDS. Protein extracts are taken up and pulled in the limai puffer, as I said before. So the protein extracts, they are taken up and then they are boiled in the limai buffer. Use the same dye, we can say Comazai for the Bradford assay. The sensitivity, it's 0.3 to the 1 microgram of proteins. Determination of the purity, molecular weight, but they are separating according to the molecular weight. The pants can be cut out for also mass spectrometry. We will talk about the mass spectrometry. It's another topic which is known as the tandem mass spectrometry. But back to the SDS page, I will share some information. Again, sodium dodecyl sulfate, polyacromide, gel electrophoresis. As we want really to know that the SDS, how it's look like, the structure. The structure is about like a fatty acid. I mean the structure of the SDS, it's like fatty acid, and in the end there is a sulfate in the center, and it's surrounded by the oxygens, and one of them also with the sodium. That is the structure actually of the SDS. So it contains hydrophobic and hydrophilic reactions. So we can say in unique detergent, I said, it carried a negative charge, and because of this it causes denaturation of this protein and give the negative charge of this protein. And again, beta mercaptoethanol break down the disulfide in the protein, and which is cannot be done by the SDS. So SDS plus beta mercaptoethanol make proteins negative charge. They all carry all the same charge, which is a negative charge, and also unfolded protein. And the gel, we use this gel just to separate the proteins from the sample. Okay, so we have first the stacking gel. The pH is 6.8, as you can see here, so it will be low pH to concentrate our proteins in one pant. And then in the separating gel the pH is 8.8, which separate our protein according to the molecular weight. Maybe the question that has come to your mind is, why is the pH of stacking gel is 6.8 while the pH in separating gel is 8.8? So you can see in the stacking gel the pH is lower, so more acidic, and that to concentrate all the proteins in one band, I mean in the stacking gel. So the purpose of the pH lower than in the separating gels, because we wanted to stacking these proteins in one band, and then we make the pH higher because we want these proteins, they are separating, they will be just migrating according to molecular weight, larger, slower, small, faster. But the question, other questions like why they are separating into the bands, why they making these proteins separating into the bands, because this gel is like a sieve, you know, small particles can pass through these pores easily while and faster, but the large moves it has to make some action to move it through these pores, so that what it makes these proteins appears like a pants on this gel, because that's why we use this gel also. We use this gel because it acts like a sieve. Small particles can pass easily and faster, while the larger particles it needs some action. That's why they are appear in the bands. It's stained by the Komizai blue, and the separation depending only in the molecular weight, because we know that the SDS and beta mercaptoethanol makes all these proteins. They are same in the size, same in the charge, and same in everything. Again, also about the SDS is the sodium to decyl sulfate, denaturation of a protein molecule, coated them with a negative charge. Beta mercaptoethanol breaks the negative charge, so they will be all, they have the same charge, have the same density, have the same shape. Only parameter left for the gel electrophoresis is the molecular weight, the size. Okay, large size, slower migration, and vice versa. There, if you hear like we hear here SDS page, is there is a native page? Yes, there is a native page, and the native page actually they are separating depending on the charge, the density, the size, the shape, because there is no SDS, there is no denaturation. So they will be separating according to the size, to the to the charge, to the dynasty, everything. So that is a special between them. The question is so important. If there is a native page and SDS page, and we feel that the SDS page is more important, or it's practical use of it, it's just to separate the proteins, we needed it. But actually we use the native page for separating of enzymes, most commonly used for separating of the enzymes. Um, and as you can see here that the SDS page, the type of the gel that we use it here is polyacrylamide gel. That's why I say PA, okay, because polyacrylamide gel. But in the case of we wanted to separate the DNA, we use agarose gel. Okay, that's also important to talk about these things. Like the type of the gel that is used here is polyacrylamide gel, and the type of the gel that we use it for the separating of the DNA is called agarose gel. And I think that's all about the gel electrophoresis here, or we can say SDS page. But I want to mention something to understand what is the Western blotting, because it will be the next topic. About the Western blot, as you can see here, that the Western, instead of this gel, we use membrane. It's called intercellulose membrane. It's called, or nitrocellulose membrane. Okay, so instead of the gel, we use nitrocellulose membrane, and that will be our topic for the Western blot, because it will be the other topic. So we had the Western blot because all our topics it's about the protein. So Western blot for separating of a protein, but there is Northern and Southern blotting. The Southern blotting for DNA and Northern blotting for the RNA. We have to know that the Western blot is for separating of the proteins, while the Northern for DNA, and the Northern for RNA, and Southern for DNA. So proteins are electrophytically transferred onto a nitrocellulose membrane, which is BVDEF, BVDF, okay, and okay, um, electroplotting on the membrane proteins are detected with specific antibodies that we use antibodies. So the other name for it is immunoplotting. We can say it's immunoplotting is the other name for the Western plot. So the plotting system, there is a tank plotting, semi-dry plotting, and the transfer buffer. The transfer buffer that contains methanol to increase the absorption of the proteins to the membranes and to remove SDS. So we use a transfer buffer. This buffer that is contains methanol, and the important that we wanted like facilitate the absorptions of these proteins to the membrane. Okay, so that why it contains the methanol, because it facilitates the absorptions of the proteins to the membrane, so we can measure it. And tank plotting large volume of the buffer, and the plotting buffer soaked in a filter paper, usually 1 hour, approximately 10 volume, which is a semi-dry plotting. So we have here the cathode, which is negative, so we can understand what is the structure of the Western plotting. Like we have here the cathode, and we have here the anode, which is the positive part. We have here filter paper, and gel, which is in the side of the negative, and membrane, which is nitrocellulose membrane, it's in the side of the positive. Detection of the plotted antibodies. This is membrane after transfer. These are the proteins. Then we do blocking, and we do the blocking by the fat milk, and after the blocking then we make the antibodies, and then second antibodies with the HRP. So we detected it by fluorescence or by the light. We can say it's labeled second antibody. So here the first antibody, and here the second antibody. We use something is called horseradish peroxidase. Horseradish peroxidase, we use it, and then that is will lead to the formation of the light cell. Why we see it's chemoluminescent, because it produces the light, so we can detect that is a protein. And sometimes it's linked to the fluorescent dye, which is not common, but actually we link it to something is called horseradish peroxidase, which is make the light. Okay, that's exactly about the Western blotting, but I have a lot of information about the Western blot, but let's focus on this in this photo because I think it's maybe helpful. So the Western blotting detect the specific proteins with the biological sample, and we said that the Western blotting we can say it's immunoblotting. So this is another name for the Western blotting, and it used for the protein extraction, protein separation, protein detection. It's not like the SDS page. We want now just make comparison between them. We want to know what is happening here. It's actually we can say SDS page is a part of the Western blotting, but the Western blotting it's not just for the protein separation. It can maybe use for the extraction and can use also for detection. So the first step SDS page is isolate the protein. Okay, how to isolate a protein from the sample picture? We use the lysis buffer, as we can mention it from the page. We said it's called lemia buffer, and then that is separating the protein from the sample. There is another buffer is called TRIPA buffer, and that is causes it's protein inhibitor, protease inhibitor, like prevent the protease from its action, and loading the gel in the SDS, the protein then separating depending on the molecular weight. The gel's form of acrylamide, it acts as a free radical, crystallize. They control the size of the pores by adjusting the concentration of acrylamide, and that may be more associated to the SDS page. What is a difference between the stacking gel and the resolving gel? So that is explain more about the SDS page, which is maybe we can say it's a part of the Western blotting. There is a stacking gel that is contains 5% of acrylamide, while the resolving gel contains 12% of acrylamide. So we can see that this resolving or separating gel, or whatever the name is, contains more acrylamide, 12% of acrylamide, because the acrylamide contains or adjusting like control the size of the pores. So we can see it's, you can see here it's the relation between the acrylamide and the pore size. Actually it's inversely, that's mean low amount of acrylamide, large pore size, while in the high acrylamide that will be small pore size, so it will be more precise. Okay, that's not our topic actually, but just we make more information about it. Like, okay, stacking gel that is contains 5% of acrylamide, so the pores they are big, really big. And then the resolving gel, it's 12% acrylamide, these pores are really small. And then we discussed all these things, but then we turn to the what is happening in the Western blot and focus on this photo. So we have these proteins and we do something what we call them as a blocking. Look, focus on this, which is known as a blocking. It's done to prevent non-specific binding of the antibodies. That's what we can say, it's a blocking here. Why we do blocking? Because there's maybe if I just put the proteins here, or antibodies, I mean, if I just put antibodies, maybe they are just bind to the not, or not my protein of interest, you know. So it's important to make a blocking, like antibodies, please don't bind to this grain area, so to prevent unspecific binding of the antibodies. What are the blocking buffer prevent non-specific binding? What are the examples? So we have two examples actually. We hear free fat milk, okay, non-fat milk, milk without any fat, and also bovine serum albumin, PSV, bovine serum albumin, bovine serum albumin. Okay, so these are the two blocking agents that we use. It is milk without fat, we can say free fat milk, and the other thing we can say it's PSV, BSA, sorry, which is a type of the albumin, and we said is a bovine serum albumin, bovine serum albumin. Then we make a primary antibody addition. Okay, we make a shaking and then wash buffer, and then we may bring the secondary antibodies, and then maybe we use two types convalescence by using the HRP, which is called horseradish peroxidase, or fluorescence, which is not more common about these things. Okay, and the horseradish peroxidase, I will talk about the horseradish peroxidase, but first I will continue about the Western blot. So transfer of these molecules, as you can see the gel is on the side of the cathode, so transfer from the gel to the membrane, to the nitrocellulose membrane, as you can see here. And the transfer that is followed by the detection of the protein, that's this is step one. There is the proteins transferred from the gel to the membrane, and then just put it here in this membrane. These things, what we call this as a blotting. Okay, why we call the Western blotting immunoblotting? Because it detects by using antibodies. That's why we said immunoblotting. So there we can see from all these steps of the Western blotting, as you can see here, that is 5 steps to detect the Western blotting. The first one, that we use a protein gel electrophoresis. This is the first step. Then we make a protein transfer. Then we're making a blocking here, antibody, first antibody probing, and detection by the secondary antibody. So actually it's composed of the protein gel electrophoresis, and then we make gel transfer. We first the protein gel electrophoresis, so we separate the protein and extract the protein from the sample, and then we make a protein transfer. Then we make blocking by the free fat milk, and we use also the bovine serum albumin, and then we make the first antibody wash, and then we make also the secondary antibody, which is labeled one, and then wash these things just to detect the protein. The membrane here that we are discussed here, the membrane, it's high affinity to the proteins and most common type nitrocellulose, as we see here, and also BVDF, which is polyurinylidene difluoride. Okay, so BVDF is polyvinyl difluoride. And why the proteins are transferring from the gel to the membrane? Good questions. Why these proteins transfer from the gel to the membrane? It's electrophoretic transfer because they are negatively charged proteins and they are just goes towards the anode. Okay. Why we use the filter paper in this condition? Because it maintains uniform flow, like make as a sandwich transfer, so the proteins not go anywhere. It will also sometimes they put a sponge which maintains a proper pressure during the transfer. And one of the component of the transfer buffers, again I see this information again, the transfer buffer is methanol, and methanol that is used as a transfer buffer because it facilitates the bindings of these proteins to this membrane. Here the buffer, the transfer buffer that we use it here, forget about the BVDF or nitrocellulose membrane. There will be a buffer here, is called methanol buffer, and this methanol buffer, it facilitates the binding of these proteins to this membrane. This is the importance of it. So remember this. Why the filter paper? Why the sponge? Because they maintain the pressure. We make it a formal transfer, etc. But the most important thing you have to know it is why we use the buffer methanol here. Why we use the transfer buffer, which is called methanol, because it facilitates the binding of the proteins to the membrane. Most used are labeled enzyme to the secondary enzymes. Yes, horseradish peroxidase, and that is makes us come, what is the horseradish peroxidase? Why we use this? Why we use this as a labeled antibodies? Because it's actually not related to the horse or radish. It's referred to the plant. It's called mirror radish. It's the enzyme actually, because we said peroxidase, and is composed of three things: peroxidase, heme protoferrin, and calcium enzyme. So it just make a reaction like oxidize the substrate using the hydrogen peroxidase, and then it produces maybe color or light, but mostly light. There are many isoenzymes of this horseradish peroxidase, but the most one is type C that we use, the most types or most isoenzyme that we use it in our experiments, type C. It large alpha helical glycoprotein which binds heme as a redox factor, so we have a chromogenic substrates and it will turn into the color products or light, and it's actually used to identify the signal. And we can say this is I will in the next lecture, because I think it's a big topic to discuss it now. So I will discuss later this topic that's about the housekeeping gene and external node OSD as Western blocking also, so you can know further information about this topic, and we end our lecture. Okay, thank you so much for listening.